Hi, my name is Mohammad Kamruzzaman. I'm a graduate student from Department of Biology at the University of Texas Arlington. My research topic is identification of gene orthologs and molecular markers for drought-tolerant nitrogen-fixing bradyrhizium strains isolated from soil in Texas. Bradyrhizium is a gram-negative soil bacteria that can do symbiosis with soybean and other legumes, and it can fix atmospheric nitrogen in the form of ammonia so that plant they can uptake it as a source of nitrogen, and in this way we don't need to provide harmful chemical nitrogen fertilizers to the environment. So in our study we wanted to do a comparative genomics with a rhizobium strain including TXV and TXEA. So TXVA, TXEA, they are isolated from Texas and they both showed better desiccation percent survivability comparing to the other rhizobium strain, and also they have shown better symbiosis and overall increase in the crop yield in the field. So for the methodology part, first we retrieve all the genome sequence that we wanted to compare, and then we first did the genome statistics just to see the overall genome properties in different rhizobium strain. For example, number of gene inside them, number of horizontally transferred gene, percent of gene GC, and number of uncharacterized gene, etc. Then we did the average nucleotide identity just to identify which genome is similar to which one based on the nucleotide sequence, and then we did the orthofinder to identify functionally similar genes in different rhizobium strain, and pangenome to identify unique genes and similar genes in different rhizobium strains. So the genome statistics result showed that the TXEA has the highest number of genes, and then the TXVA, and then USDA 110, which is a novel rhizobia strain, and USDA 110 has the highest percent of GC, and USDA 110 also has the highest number of horizontally transferred gene. Based on average nucleotide identity result, we found that both TXVA and TXEA, they are highly similar to each other, and also they are highly similar to Bradyrhizium japonicum strain E109, but also they are highly dissimilar to CC275E strain. For gene ortholog study, we followed orthofinder pipeline where we get different information like orthologs, orthogroup, then gene duplication event, and from that data set we actually produce this phylogenetic tree. And for example, this A actually showing the rooted gene phylogenetic tree for one single gene, B here is showing the gene duplication event, the number of gene duplication event for different rhizobium strains, and C actually the rooted species tree. So for the pangenome analysis, we followed the Rory pipeline, and after the analysis, it produced a heat map of pangenome matrix that actually showing the number of gene cluster. The gene cluster that contains different number of genes that are similar, that are common in different rhizobium strains, and that are some of them cluster are actually unique for each rhizobium strains. So once we have the list of unique genes, we did the blast in the NCBI database just to make sure these genes are not available in any of the species in the database. So based on NCBI blast, we've found that one gene for TXEA and one gene for TXVA that has no match overall with any of the species and any of the strains sequence. Afterward, we did the PCR to amplify the genes from TXVA and TXEA. So in conclusion, RPSJ, this is the gene we found as a core gene in all compared genomes, and this gene actually responsible for translation, ribosomal structure, and biogenesis. And this deoxycytidyl deaminase gene, we found this gene as a unique gene for TXEA, and hypothetical protein, this gene found unique for TXVA. The take-home message from this research is we can apply these tools in genome comparison for newly sequenced genome. We can functionally identify the unique genes that were not studied before, and we can also use this unique gene as a molecular marker to identify rhizobia in the soybean field. Thank you.